And welcome to the third after that fourth session of the power hour. And today we are going to discuss very important chapter, second chapter of genetics, that is the molecular basis of inheritance. And as I told you in previous lecture also, this chapter counts nearly 24 marks, means previously 6 MCQ going to ask from this chapter. So in today's session, we are going to discuss some important topics, some important concept of this chapter. First subtopic is structure of DNA. Second subtopic is packaging of DNA, and some important MCQ, only MCQ of protein synthesis. Protein synthesis, you know that there are two processes. One is the transcription, second one is the translation. The conversion of DNA into mRNA is a transcription, and mRNA into protein is known as the translation. This complete process is known as the central dogma as well as protein synthesis. Only MCQ we are going to discuss in that particular topic. So in detail we are going to discuss structure of DNA, packaging of DNA, and for MCQ we are going to discuss only protein synthesis, that is the transcription and translation. So let's start this fourth session of power hour with the topic structure of DNA. And if you know the structure of DNA, now here there are some points which is give us the information, little bit input about the DNA. So first input is Friedrich Miescher, the scientist F. Miescher, on 1869. In 1869, identified DNA and name as nuclein. Because the concept you have to understand, there is a cell having nucleus, and nucleus having some substance. In today's date, if I am asking you what is the genetic material present into the nucleus, your answer may be DNA. But in 1869, no one knows about the DNA. So he isolate one substance from nucleus. This substance which is isolated from the nucleus is given name as nuclein because it is isolated from nucleus. After some days, like after 20 years, this substance which is the nuclein, on 1889, scientist named as Altman found that this nuclein have some acidic property. This nuclein have some acidic property. This nuclein have some acidic property. So now in today's date, we call it as a nucleic acid. We call it as a nucleic acid, and nucleic acid are of two types. One is a deoxyribonucleic acid, and second one is the ribonucleic acid. Deoxyribonucleic acid is known as DNA, and ribonucleic acid is known as RNA. So 1869, Friedrich Miescher identified DNA and named it as a nuclein, and James Watson and Francis Crick, in 1953 proposed the double helix model of DNA. So now this model is based on the discovery of Wilkins and Franklin, Rosalind Franklin and Wilkins. So this model of given by Francis Crick and James Watson is depend on Rosalind Franklin's model and Wilkins model. Okay, so now let's going to discuss about, first of all, we are going to customize the slide, and let's going to discuss about the structure of DNA. I am giving here a title. That is a structure of DNA. Structure of DNA. So this is the title, and the structure of DNA topic we are going to discuss in today. So now understood first line. I am writing here, DNA is a polymer of nucleotide. My first line mentioning here, DNA is a polymer of nucleotide. So for study a DNA, we have to understand how nucleic acid or nucleotide look like. So second point I am writing here, second point, nucleotide equal to is equal to pentose sugar plus N2 base means nitrogen base, and third component phosphoric acid. Third component is the phosphoric acid. So when phosphoric acid, nitrogen base, pentose sugar combines together with different bond, which I am going to discuss in further slides, the combinedly there is a formation of nucleotide, and long polymer of nucleotide responsible for formation of one strand of DNA, and other long chain of nucleotide responsible formation of second strand of DNA, and two strand of DNA close to each other, comes close to each other, having attraction, having helical structure known as DNA. Now, if I am talking about the nucleotide, pentose sugar, nitrogen base, phosphoric acid. So how this pentose sugar looks like? Pentose sugar are having two type. One is the deoxyribose, and second one is the ribose. One is the deoxyribose and second one is the ribose. So when we talk about the deoxyribose, it means there is 1, 2, 3, 4, 5. Now counting of the carbon is like, this is the first carbon, this is second carbon, third carbon, fourth carbon. Where is fifth carbon? Where is fifth carbon? Fifth carbon is here. Now this is this one is fifth carbon. Now on first carbon, here is H and OH. On second carbon, it is having H and H. On third carbon, H and OH. Fourth carbon, only one hydrogen here, CH2OH. Now when we talk about the ribose sugar, because here is a deoxyribose sugar and it is a ribose sugar. So when we draw the structure of ribose sugar, it look like this. Oxygen 1, 2, 3, 4, 5. Everything same. Like on first carbon, H, OH. On third carbon, H, OH. On fourth carbon, H. On fifth carbon, CH2OH. Everything is same. On second position, observe that in deoxyribose there is a HH. And in case of ribose sugar, here is a H and OH. One extra functional group present in the ribose. So now on the basis of the structure, can you tell me which one is stable? Deoxyribose is stable or ribose is stable? On the basis of this, can you tell me which one is stable? Deoxyribose or ribose? Anyone? Anyone from class? Look, logic is very simple. Which of the following having maximum functional group? So there is one functional group here, second one, third one, fourth one, maximum functional group in the ribose, and one lesser functional group, one alcoholic group is not present on the second position in case of deoxyribose. It means deoxyribose is not that reactive and ribose is reactive sugar. So the sugar which is reactive having less stability. So ribose is reactive, it means it is less stable. Deoxyribose is having less functional group, it means less reactive, means more stable. So that's why we human have a genetic material in the form of DNA. And in case of viruses, virus is not that developed, so some viruses have genetic material in the form of RNA due to stability. And this stability, one of the reason of the stability is the deoxyribose sugar and ribose sugar. So I hope so nucleotide's first component, that is the pentose sugar, you understood here. Here is a deoxyribose and here is a ribose. Second component is a nitrogen base. Second component is a nitrogen base, and nitrogen base is divided into two types. One is purines and second one is pyrimidines. One is purine and second one is the pyrimidine. Purine have two different further types. Purine have two different types and pyrimidine have three different types. I will tell you which one is present in the DNA and which one is present in the RNA. So now, when we talk about the purine, adenine and guanine are the purine, and thymine, cytosine and uracil are the pyrimidine. But when we talk about the DNA, adenine and guanine, purine is common in DNA and RNA. But when we talk about the pyrimidine, T with C present in case of DNA and C with U present in case of RNA. So we can say that T is replaced by U, means thymine is replaced by uracil in case of RNA. In case of RNA, and 5-methyl-uracil is also known as thymine. 5-methyl-uracil is also known as thymine. This is the second reason why DNA is more stable, because it has thymine instead of uracil, and thymine is a modified version of uracil. So second reason, because of DNA is stable and we are having genetic material in the form of DNA. So now when we talk about those purine and pyrimidine, little bit we can say that in a detailed manner. I am going to write here something. This is at one point. Now one point I am writing here, purine is dicyclic and pyrimidine is monocyclic. Purine has nitrogen, have N2 at first, third, seventh and the ninth position. It has nitrogen on first, third, seventh and ninth position. And in monocyclic pyrimidine, it have nitrogen at first and third position. Those are the detailed thing. Now directly coming to the third category, that is known as phosphoric acid. Phosphoric acid formula is very simple, H3 or PO4. H3PO4. Now there is one phosphorus and all four direction we are giving oxygen, 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 oxygen. How many hydrogen are there? 3 hydrogen, OH, OH, OH. Now for that one oxygen, here is double bond. This is a phosphoric acid. Now this is separate phosphoric acid, this is a separate nitrogen base, and this is separate about the pentose sugar. Which one is present in DNA? If we talking about the deoxyribose or ribose, in case of DNA, deoxyribose is present. Which is present in the DNA? Purine or pyrimidine, both are present. Purine, adenine and guanine. And what about pyrimidine? DNA have thymine and cytosine. Uracil is not present in case of DNA. Now phosphoric acid, it is present in the DNA. Now let's draw one nucleotide first, because Thus, 3 are the component of nucleotide, and nucleotide combine together to form a DNA. So first of all, we have to assemble those 3 component. Then after, with the help of these 3 component, we are going to manufacture the nucleotide. With the help of long nucleotide, we are going to synthesize the DNA. Let's start. I am adding one slide here, and let's copy that slide to understand previous thing also. I am taking this at one corner for my reference. Now we are having this kind of thing. Let's start with the further thing. We are having those all component. Now I want to draw one nucleotide. Nucleotide. So for drawing nucleotide, I have to draw here one pentose sugar, HOH, here HH, here is HOH, here is H on 5th number carbon, CH2OH. Now, at which position nitrogen base is attached? On first. So here, bond formation takes place. And N2 base is attached. It will be adenine or guanine or thymine or cytosine. Not uracil, because we are talking about the DNA now. On 5th position, this is the 5th position. Here pentose sugar is attached. Not pentose sugar, sorry. At 5th position, phosphorus group is attached. Phosphorus, here is OH, here is OH, here is OH, and double bond O. So, elimination of water, this hydrogen, and there is an elimination of H2O, formation of bond here, and this bond, 2 bonds are formed here. One bond is known as glycosidic bond, and second bond is known as phosphoester bond. Now I am not talking about the phosphodiester bond. Further, in case of DNA, we are going to draw phosphodiester also, but here only one ester bond is formed because of release of one H2O molecule. So here is phosphoester bond. Diester bond, I will tell you where it is going to be formed. So this is the structure of nucleotide. The long chain of nucleotide is responsible for formation of, yes, one DNA strand. Let's check. In second slide, here I am going to draw one pentose sugar having on first position nitrogen base. Guess any nitrogen base. For example, I am guessing here adenine. And on 5th position, they are having phosphorus group is attached. Now second nucleotide I am drawing here. Second nucleotide. On first position, for example, thymine. And on 5th position, phosphorus. Now third, here there is a pentose sugar. On first position, there is guanine. Here, phosphorus. After that, 1, 2, 3, fourth position, there is cytosine, and here phosphorus is present. Another we are going to draw here. Here is, let's see, guanine. Here is phosphorus. Now this is the long chain of nucleotide, but there is no bonding. There is no linkage between the nucleotide. So only one functional group is present here for this linkage. This OH, this remaining OH. Now the bond is formed between the third carbon of ribose sugar, deoxyribose sugar, and phosphorus. Now this bond, this is the phosphoester bond. but this bond, 2-ester group is released. So that's why this bond is known as the phosphodiester bond. This bond is known as phosphodiester bond. This bond is known as phosphodiester bond. This bond is known as phosphodiester bond. So this is the one strand of DNA. Only one strand of DNA. How many of you are joined here? 4 students are watching. Guys, you are getting my point. The students are watching the streaming, live streaming. You are getting my point? Yes or no? Please write in the chat section if you are getting my point. Clear? I draw here one strand of DNA only. I'll clear other doubt also. Just give me a thumb if you understood this much of the topic. Anyone? Yes, pro number 1 is saying yes sir, okay. Let's discuss further. So this is the only one strand of DNA. Now, guys, understood how come 5' end and 3' end are? Now, this is phosphorus is attached at 5' end, and here at the end 3' end OH is free. So because of this OH is free here, it is given as a 3' end. And here there is a no free, but phosphorus is attached as 5'. So here is opposite is 5' end. Like the same way, I am drawing on other side also. This is a pentose sugar. So now this is the first carbon, and this first carbon is attached with guanine here. So guanine is always paired with cytosine with the help of triple bond. And on 5th carbon there is a phosphorus. Understood this thing? I will clear it one more time. Look, this is the oxygen. First carbon, second carbon, third carbon, fourth carbon, fifth carbon. Fifth carbon have phosphorus. And first carbon have guanine having triple bond because cytosine is always paired with guanine. Now, one another. Understood the thing? Oxygen 1, 2, 3, 4, 5 and this 5th. Phosphorus, first carbon, guanine, cytosine with triple bond. Now, above side 1, 2, 3, 4, 5. Here is phosphorus. Here adenine. Now, in between adenine and thymine there is double bond. Lastly, here is oxygen 1, 2, 3, 4, 5, phosphorus, thymine, double bond. So now we have to join this nucleotide also because 3' with phosphorus, 3' with phosphorus. This is a phosphodiester bond, phosphodiester bond. Now, on the basis of that structure, OH group is free. OH group is free here. So third position 3' end and opposite one is 5' end. Opposite one is 5' end. If I draw this structure like a DNA strand, so if I draw this structure as a one DNA strand and this is another DNA strand, so let's give a name a 5' end and 3' end, 3' end and 5' end. Here is adenine, thymine, guanine, cytosine, guanine. A, T, G, C, G. On other side, on other hand, here is T. In between double bond, A, in between double bond, C, triple bond, G, triple bond, C, triple bond. This is a DNA, but is DNA is looking like this? No. When we hold this like this is DNA. If I am holding this and twisted, so twisted ladder-like structure we have to draw here. So this structure is somewhat looks like, here is one strand of DNA, and I am drawing another strand here. Now, this one is 5 prime end, 3 prime end, 3 prime end, and 5 prime end. If we talk about the diameter of DNA, then it is a 20 angstrom. Diameter of DNA is 20 angstrom. There is a nitrogenous base present here, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20. Like that nitrogen base is present. Here, this groove is known as major groove, and this groove is known as minor. Major, minor. Alternate major and minor groove is present. I am writing here major groove, and this one is minor groove. This one is major, minor. So alternate major and minor groove are present. So understood the thing. We have to calculate one 360-degree turn, major to major or minor to minor. If I am talking about major groove to major groove, means this is a 360-degree turn. This much is a 360-degree turn. This much is known as 360-degree turn. 360-degree turn. 360-degree turn. In that 360-degree turn, there is 10 base pair in case of bDNA. There are type of DNA: A DNA, B DNA, C DNA, D DNA, not E DNA, Z DNA, A, B, C, D, and Z DNA. So the DNA model which is studied by Watson and Crick, it is a bDNA. It is type of bDNA. And if I am talking about the bDNA, here I draw the structure of bDNA. This bDNA have 10 base pair. In 360-degree turn. And how we calculate 360-degree turn? Major to major groove or minor to minor groove. How many? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. How many base pair? 10 base pair. So if I am talking about the distance of this 10 base pair, now the 10 base pair, now this 10 base pair have a distance of 34 angstrom. How much? 34 angstrom. So the calculation is very simple. 20 angstrom means 2 nanometer. 34 angstrom means 3.4 nanometer. Now, if we talking about the distance between two consecutive base pair, if we are talking about the distance between two consecutive base pair, 10 base pair divided by 10, it means how many? How much? 3.4 angstrom and 0.34 nanometer. This is the distance, and this is the structure of DNA according to Watson and Crick. In next slide, they are given us the various information. DNA is made up of 2 polynucleotide chain. Yes, there is one polynucleotide chain. Second polynucleotide, 1, second, 1 and second. It is right-handed fashion because it is a bDNA. Only ZDNA, for the information you have to understand, only ZDNA have anti-clockwise or left-handed fashion. Otherwise DNA A, DNA B, DNA C, DNA D having which pattern? Right-handed pattern. Pitch of the helix having 3.4 nanometer or 34. What is the pitch? This 360-degree turn. What pitch means? 360-degree turn. Number of base pair in each turn is 10. Already we discussed. The distance between adjacent, matlab one after another. Adjacent means one after another. There is 0.34 nanometer or 3.4 angstrom. I hope so you understood this topic. Now backbone is sugar and phosphate, running anti-parallel, that 5 prime, 3 prime, we already discussed. Now there is one question for you. One question for you. How many students are there in a class? Mayank, pro number 1. Guys, question for you. The question is very simple. Question is not that difficult. Question is very simple. Question is asking you, if the distance between two consecutive base pair is 0.34. What is the meaning of question? If the distance between two consecutive base pair is 0.34 nanometer and the total number of base pair of the DNA double helix in mammalian cell is 6.6 into 10 to the power 9, then the length of DNA is approximately. Then length of DNA is approximately. Length of DNA is equal to the distance between one base pair into total base pair. The distance between one base pair is 0.34 nanometer. Nanometer into meter is into 10 to the power 9. To the power 9. Yes or no? Yes. Now, if we calculate, then 6.6 into 10 to the power 9 base pair. 10 to the power 9 base pair. Nanometer to meter is minus, so this cancels. So 0.34 into 6.6, it is 10 to the power minus 9, because we are converting nanometer into meter. So 0.34 into 10 to the power minus 9. So minus 9 plus 9 cancels, 6.6 into 0.34. Let's calculate this. We have tool here. We can use the tool easily. Tools. We have tools. Okay. Maybe in NEET we have to take a tool, disciplinary tool. In case of math we can find calculator, and here is calculator. So we have to calculate here 0.34 multiply by 6.6, 2.244. It means 2.244 is nearly equal to 2.2, because we take SI unit, so meter, so 2.2 meter. Any answer 2.5, 2.0, 2.7, 2.2, D is correct answer. This is the solution. What we did? Length of DNA is equal to base pair into total base pair. Complete DNA length we calculate. Now, there are two major rules. One is base pair rule. Second one is the Chargaff rule. We are going to discuss this rule with the help of a blank slide. Let's add one blank slide. One rule is base pair rule. Base pair rule. What it say? Purine. The statements of base pair rule is purine always pair with pyrimidine. They are saying purine always pair with pyrimidine. It means purines are A and G, and pyrimidine in case of DNA is T and C. So A and T always form pair. A is not able to form pair with C. A is only able to form pair with T with the help of double bond. C always form pair with G with the help of triple bond, or vice versa, T double bond A or C triple bond G. This is base pair rule. Second one is the Chargaff's rule. What Chargaff's rule told us, we are having Chargaff rule with us. This is the Erwin Chargaff which is giving us the rule. They are saying number or proportion of adenine equal to T, means now A equal to T and G equal to C. It means now I am not drawing the bond. A equal to T and G equal to C. It means A divided by T equal to 1 and G divided by C equal to 1. Another meaning of this Chargaff rule is A plus G is always equal to T plus C. It means A plus G upon T plus C equal to 1. Another one meaning of this Chargaff rule is A plus G plus T plus C equal to 100% of DNA. A plus G plus T plus C equal to 100% of DNA is nitrogen base. Now on the basis of that, can we have one question? Yes, we have one question. Here they are saying if adenine makes 30% of DNA molecule, what will be the percentage of thymine, guanine, cytosine in it? You have 1 minute of time to solve this question. I am taking water and you have 1 minute of time to solve this question. Let's try to solve this question. This is easy one, but let's now A equal to T. A equal to T. It means adenine are 30. So thymine is also 30. So thymine 30. Any option, only one option is there. But we have to calculate the possibility of guanine and cytosine also. So A plus G plus T plus C equal to 100%. Now A is given 30%. We already calculate T 30%. Let's consider guanine as a X and cytosine as a X because both having proportion similar. Guanine and cytosine is equal to 100%. So 2x plus 60% equal to 100%. Now 2x equal to 100% minus 60%. So 2x equal to, here is 40%, x equal to 40% divided by 2, x equal to 20%. It means guanine is equal to 20% and cytosine is equal to 20%. Thymine 30, 30, 20, 20. 30, 20, 20. C is correct answer. Let's move to the Another some important point, this is theoretical point, we have to remember this point, that 5137, a bacteriophages, it is a virus that affect the bacteria, has 5386 nucleotide, very small. Now bacteriophages lambda, it is little bit developed as compared to the 5174. 48,502, it is looking like the government seat of MBBS, okay. 48,502 base pair in case of bacteriophage lambda. E. coli, it is not a virus, it is a bacterium. It has 4.6 into 10 raised to the power 6 base pair. And haploid content, haploid means N. Haploid content of human DNA is 3.3 into 10 raised to the power 9, and 2n is equal to 6.6 into 10 raised to the power 9. This is the data, we have to remember this data. Now length of DNA equal to number of base into distance between two consecutive base pair. Just before some time we discussed it. Length of DNA, length of DNA is equal to number of base pair. So diploid have 6.6 into 10 raised to the power 9 into distance between two consecutive base pair is 0.34 nanometer, means 10 raised to the power minus 9 meter. Cancel. 6.6 into 0.34 equal to 6.6 into 2.2 meter. We already calculated that. Now packaging of DNA in eukaryote only we are going to discuss. Now if we are saying packaging, why there is a need of packaging? Just before the slide we discussed the length of DNA is 2.2 meter. The length of DNA is 2.2 meter. And if I am asking you what is the size of cell, now I am asking you what is the size of nucleus, your answer is, sir, 10 raised to the power minus 6. Now this small nucleus and this much long DNA, how come it is coming or compactly arranged into that small nucleus? It is due to the packaging. It is due to the packaging. Now what we have to discuss in packaging of DNA? In eukaryote there is a set of positively charged protein. Protein is always positively charged and DNA is negatively charged. So positively charged protein is called as histone, and histone rich in two type of amino acid. We have to remember this name. In NEET examination they are going to ask you lysine and arginine. Lysine and arginine. Now this complete structure, histone, octamer, nucleosome, I am going to discuss with you with the help of this blank slide. Now this is a topic, packaging of DNA, and here packaging of DNA. Now this part, first of all, histone protein, you know that how many histone? 4 histone, H2A, H2B, H3, H4 multiply by 4. Not 4, multiply by 2 equal to 8, because octamer, no? So for octamer we have to multiply by 2. So 8 histone protein. How it is arranged? Now for example, this is H2A, this is H2B, this one is H3, and this one is H4. On above side or back side, here H2A, here H2B, H3, H4. This is the histone octamer. What is the charge of this octamer? Positive charge. What is the charge of this octamer? Is positive charge. Now on that positively charged protein, there is a wrapping of DNA, 1/3 and here. So the DNA, this is the DNA. Charge of DNA is negative. So if I am talking about this DNA, the DNA which wrap around the complex of histone octamer, this is known as the core DNA. Core DNA. And this DNA which is present here before the rounding or wrapping of octamer, this is known as the linker DNA. Core DNA have base pair of 146 base pair and linker DNA have 54 base pair. So combined total is 200 base pair. One nucleosome, because here is octamer, but after wrapping of DNA, it is called as nucleosome. It is called as nucleosome. So now, this is for example one nucleosome, second, third, fourth, fifth, sixth, and this is DNA. Coming, one wrap, going, one, Now how many turn? If in NEET exam they are asking you how many turn it took to wrapping histone octamer. So this is known as 1/3/4 turn, or we can say that 1.75 turn. 1.75 turn or 1/3/4 turn. Now second question. This is the 1, 2, 3, 4, 5, 6. How many nucleosomes are present? On the basis of this, means if we know the distance of DNA, we can easily calculate, or we know the base pair of DNA, we can easily calculate the, what we can say, nucleosome, because divided by complete dose base pair divided by 200 is total nucleosome. So this negatively charged DNA wrap and positively charged protein, histone protein present in middle. So this is complete structure of one structure. This is known as the beads on string structure, and thus beads on string structure is known as the chromatin fibers, and in the form of chromatin fiber DNA is packed. Now after that, this chromatin fibers we can pack inside the chromosome, and in DNA chromatin fiber or chromosome is present, or we can say that in case of in nucleus, this chromatin fiber or chromosome present in the form of genetic material. So this is the chromatin fiber. This is the DNA wrap around the, what we can say, nucleosome. This is the complete packaging of DNA, and because of this packaging, it is easy to pack or compact 2.2 meter of DNA inside the small nucleus. Now, coming to the next point, a typical nucleosome contain how many base pair? 200. How we calculate this base pair? Core DNA have 146. Linker DNA have 54. 146 plus 54, 200 base pair. Therefore, the total number of nucleosome in human, if we have to calculate the total number of nucleosome in human, we know that a total number of base pair in human. So 6.6 into 10 to the power 9 divided by 200, right? So this cuts 7, 3.3 into 10 to the power 7, thus amount of nucleosome. So beads on string structure, we already discussed this. Chromatin fiber packed into the chromatin fiber. Chromatin is packed, chromatin fiber coiled and condensed and metaphase stage, it is packed inside the chromosome. And higher level of packaging of chromatin required non-histone protein also, like this is the chromosome. Here is a histone protein and chromatin fibers. Now we need some non-histone protein also here to pack this chromatin fiber in the chromosome, and it is observed in a stage of metaphase of karyokinesis. Now, there are two types of chromatin, euchromatin and heterochromatin. Euchromatin is loosely packed. If it is loosely packed, it means it is a transcriptionally active. Second one is the heterochromatin. It is a densely packed. Densely packed means transcriptionally inactive. Those are the 2 points. First question, if the length of DNA, they are giving us the length of DNA 1.1 meter, what will be the approximate number of base pair? Very easy question. How can we calculate? If for 2.2 meter, how many base pair? We already calculated in previous slide. For 2.2 meter, base pair is 6.6 into 10 raised to power 9, right? For 2.2 meter, base pair is 6.6 into 10 raised to the power 9. So exactly half, 1.1 meter. So answer will be 3.3 into 10 raised to the power 9 base pair. Any option? 3.3? No. 3.10 raised to power 9. Here is just minute difference, 10 raised to power 6 and 10 raised to power 9. We need 10 raised to power 9. Next question. Oh, this is a complicated question. Just look at the question. Read the question. Read the following question. There are 5 statements and they are asking us about the correct statement. So 5 statements and maybe 3 are correct. Out of those 5, 3 are correct. First, euchromatin is loosely packed chromatin. Yes, it is correct. Just before 2 seconds, we read this. Heterochromatin is transcriptionally active. Wrong. It is inactive because it is compactly packed. Histone octamer is wrapped by negatively charged DNA in the nucleosome. It is correct, right? Just we discuss in case of packaging. Histone are rich in lysine and arginine. I think it is also right. A typical nucleosome contains 400 base. Nucleosome have 200 base pair. If they are if they are saying bases, then 200, 200, 400 bases. But they are saying base pair. Base pair means only 200 be required. So because of this, this is statement false. So A, C and D. A, C, D. Option number A is correct. A, C, D. Good question. According to NEET point of view. Second question. Now, just I told you in first 5 minutes, we are going to discuss structure of DNA and packaging of DNA as well as question. But for protein synthesis, I am going to discuss only MCQ because we have very less time. In 1 hour, I am trying to cover almost MCQs. The process of translation. Translation is mRNA to protein. mRNA to protein. Begin as soon as the large subunit, no. Both subunit, no. tRNA activated, no. Small unit encounter mRNA, any option. Small subunit of ribosome encounter mRNA, D. Which of the following RNA is not required for the synthesis of protein? There are 3 RNA, mRNA, rRNA, tRNA. All 3 required for protein synthesis. So mRNA required, R required, T required. This RNA, siRNA, no. This is calculation based question. Just look at the question. E. coli bacteria has 4.6 into 10 raised to power 6 base pair. Okay, they are saying base pair. And complete the process of replication in 18 minutes. Okay, the process of replication they are completing in 18 minutes. Means, okay. Then the average rate of polymerization is approximately, okay, per second. So 18 minute equal to how many second? 18 into 60, right? So 18 into 16, 1080 second, right? And total base pair are 4.6 into 10 to the power 6 upon 1080. The answer will be, means the answer of this solution is our answer. Let's calculate with the help of calculator because I am not a mathematics teacher and my calculation is not that strong, but in NEET examination you don't have calculator, so calculate by your method only. So 4, 6, 5, let's see, 5046. How to erase it? 4, 6, 1, 2, 3, 4, 5 divided by 1080 is equal to 4259. 4259. Any option 4259? 4259. There is no option 4259. But they are asking approximately, no? If they are asking approximately, it means not accurate answer they are given. Approximately. So 4259 is approximately not 2000, 3000, 4000. 4000 base pair per second is correct answer. The actual answer is 4259, but they are asking approximately, so that's why 4000 base pair per second is the correct answer. Now next question, which is the only enzyme that have capability to catalyze initiation, elongation, termination in the process of transcription? Transcription is DNA to RNA. So we need RNA polymerase. Which RNA polymerase? Because DNA to RNA, DNA dependent RNA polymerase. Any option DNA dependent RNA polymerase, B number option. Let's check another MCQ. What is the role of RNA polymerase 3? So understood the concept first. If we are saying RNA polymerase 1, then it is responsible formation of rRNA. If I am saying RNA polymerase 2, it is responsible formation of hnRNA or mRNA. It is converted into mRNA after some time, after process. RNA polymerase 3 is responsible for formation of tRNA. So they are asking 3, then it is a tRNA. tRNA, 5S and RNA, yes, B. B is the correct. Next, the name of enzyme that facilitate opening of DNA. It is related to replication. Opening of DNA is DNA helicase. So this is the correct answer. Next, in the process of transcription in eukaryote, the RNA transcription in eukaryote. Now what is transcription? Transcription is conversion of DNA into RNA. It will be rRNA, tRNA or mRNA. But there are 3 types of different RNA. RNA polymerase 1, which is responsible for formation of, give me the exact answer. RNA polymerase 1 is responsible for formation of, just the second before I told you, RNA polymerase 1 is responsible for formation of rRNA. RNA polymerase 2 is responsible for formation of hnRNA. RNA polymerase 3 is responsible for formation of tRNA. Now they are asking us about RNA polymerase 1. So I think so rRNA is correct answer. rRNA is correct answer. Any option? mRNA with additional process? No. tRNA is no. rRNA with 28 years, 18 years and 5.8 years? Yes. Precursor of mRNA? No. C is correct answer. rRNA is. Now, last question of the day. We have last 2 minutes and last question of the day. Let's discuss, match the following. How many of you are in the session? Okay. Let's start discussion of last MCQ of this. RNA polymerase 1, RNA polymerase 2, RNA polymerase 3. We know that RNA polymerase 1 is for rRNA, right? rRNA. RNA polymerase 2 is for hnRNA, or we can say that mRNA, because mRNA is a processed RNA, capping, tailing, slicing, splicing and all those things. So, but formation of hnRNA is takes place. So RNA polymerase 2, RNA polymerase 3, tRNA. Now, on the basis of that, RNA polymerase 1 is 2, A2 is only in one option. Let's just check. Second is 3, means B3 present here, but It is not correct answer. RNA polymer is 3-1, 3-1, 3-1, 3-1, 3-1, C1 here and here also. It is a correct match. C is a correct answer of this question. I hope so. You understood molecular basis of inheritance in detailed manner. I tried my level best to draw the structure of the DNA, to explain packaging of DNA in very short and precise manner. Some question of transcription, translation and protein synthesis I have discussed in today's session. I hope so you understood this session, and in next session we are going to discuss some more topics of botany. Let's ensure your presence in another upcoming session. Thank you from my side from today's session. Bye bye and take care. All the best for your NEET examination.